Hello everyone, welcome again to Supplement Tutorials. In our previous microbiology video, we talked about the internal components of the bacteria. Today we are going to talk about the classification of bacteria, and we are going to classify bacteria based on a number of categories. Alright, so let's get started. Alright, so we can classify bacteria based on their cell wall. We can also classify bacteria based on their morphology. We can classify based them on atmospheric requirement. We can also classify them based on temperature requirement. We can also further classify them based on their nutritional requirements. And sometimes based on their pathogenicity. Alright, so pretty quick, let's talk about their cell wall. Okay, so bacteria cell wall allows them to be classified into two categories. We have the gram positives, and then we have the gram negative. Okay, so in our previous videos, we got to realize that the gram-positive bacteria are the bacteria that have a very thick layer of the peptidoglycan, and as a result, it's able to retain the crystal violets of the gram stain, so therefore appears to be seen as blue to purple color under the microscope after it has been stained, or after it has undergone gram staining. And then in our video that talked about the structure and then the function of the bacteria, we realized that the gram-positive bacteria have their cell wall has certain components that differ from that of the gram-negative bacteria. And what we established was that the gram-negative bacteria has a very thin peptidoglycan layer and therefore does not retain enough of the crystal violet. So in the decolorization process of gram staining, it is washed off. That is, the crystal violet component is washed off, and then it then what absorbs the counter stain, that is safranin. And the safranin appears to be pink to red. So under the microscope, we would see that the gram-negative bacteria appears to be pink, or appears to assume the color of pink to red. Alright, so that is basically how we can distinguish the bacteria based on their cell wall. If you want to know more about the gram-positive and the gram-negative bacteria, you can visit our previous videos. We have talked about these two. Alright, now we can give some examples of the gram-positive bacteria. So we can talk about the Staphylococcus. We can talk about the Streptococcus, and so on. And then for the gram-negative bacteria, we can talk about all members as let's see, most members of the Enterobacteriaceae species. Most members of the Enterobacteriaceae Enterobacteriaceae family, members of the Enterobacteriaceae. So we can talk about Escherichia coli, we can talk about Pseudomonas aeruginosa, we can talk about a lot of them, Helicobacter pylori, etc. Alright, now let's talk about the classification of bacteria based on their morphology. So based on morphology. So morphologically, we can classify bacteria into cocci. We can classify them into bacilli. We can also classify them to spherocytes, or let's say spirals. Okay, so basically the bacilli, the cocci, are actually spherical in shape. They are spherical in shape. So the cocci are spherical in shape. Most of the cocci are often gram-positive bacteria. Then we have the bacilli. The bacilli are rod-like in shape, like this. They are rods. They are also called rods. Okay, and most of them too are gram-negative bacteria. And then the spherocytes, they often assume a spiral shape. So some of them, and their spiral shape can vary. Some of them appear like this, like a comma shape. That is the vibros. We also have those that are like this, like the Treponema pallidum, or the causative organism for syphilis. So that is how they appear. And this one is the vibros shape, or the vibrocorrelate. Yeah. Example of the vibros shape is the vibrocorrelate. That's the causative agent for cholera. So we have the cocci. So under cocci, we can talk about the Staphylococcus. So the Staphylococcus, they are actually cocci that have clustered. So they often appears like this under the microscope. So like this. Okay. So these ones are Staphylococcus. Okay. So they appear in clusters. Alright. So examples, we can talk about the Staphylococcus aureus. Staphylococcus aureus. Alright. Okay, we can also talk about the Micrococcus. Alright. Okay. Under the cocci, we have organisms that are diplococci. So the diplococci die means two. So they are in pairs like this. They are in pairs. So they are cocci that are mostly found in pairs. So that is the diplococcus. Then we have the tetras. So the tetras, they are actually in forms. So they appear like this under the microscope. So this one is the tetras. They appear in four forms. An example is the micrococcus. So we can say that the micrococcus is an example of staphylococcus, and it's also a tetrad. An example of the staphylococcus because it is arranged in clusters, and it's a tetrad because it's in four. Then we have the octads. Octads, they are in eight. They are a group of 8 clustered cocci. That is the octads. Alright. Then aside staphylococcus, we also have, still under the same coccus, we also have streptococcus. So they are often cocci that are arranged in chains. So you can see them like this. So they are what? They are streptococcus. Okay. So examples of the streptococcus, we have the streptococcus pyogenes. Then we have streptococcus pneumoniae. Then we have the different types. We have the Streptococcus agalacea, we have the S. agalacea, etc., etc. So that is the Streptococcus. Okay. All right. So now let's talk about the bacilli. So the bacilli are mostly rods. They are rods. So mostly they appear in a rod form under the microscope, and most of them are gram-negative bacteria. So they are mostly rod forms. So most members of the Enterobacteriaceae, most members of the Enterobacteriaceae. So when we talk about the Enterobacteriaceae, we can talk about the E. coli. We can talk about the Proteus mirabilis. We can talk about Helicobacter pylori. We can talk about the Campylobacter. We can talk about the Pseudomonas aeruginosa, etc. So most members of the Enterobacteriaceae, they are rods. Okay. So those are the bacilli. Now we have Streptobacilli. So strepto in chains, and then bacilli, they are rods. So anytime you see rods that are in chain, they are what? They are streptobacilli. Alright. Anytime you see rods, see like this under the microscope, you see rods that are in chains, they are what? Streptobacilli. And there are also bacilli that are arranged in what? In palisades form. So they are arranged like this. They are like clusters, but they are together like this in palisade form. Okay. So that is also a kind of what? Bacilli that we have. Alright. Then we have a short curved form of the bacilli. It's like a coccus and it's like a bacilli. So those ones are called cocobacilli. So we have the cocobacilli. And those ones, they are short forms of the bacilli. They are short curved or curvy forms of what? Of the bacilli. Alright. So those are the cocobacilli too as well. So that will be for the bacilli. Then we have the spherochaetes. Or the spirals. So those ones, they are like spiral forms. Some of them are like this. You can have the comma shape like the vibro. You can also have the irregular spiral shape like the Treponema palladium. So typical examples of these ones are the vibros. We have the vibro correlate. Then we have the Treponema palladium. That is the causative agent for what do we call it? Syphilis. So those are what? Spherochaetes or spirals. Alright. Now we are going to classify the bacteria based on their. Now we are done with their cell wall, we are done with their morphology. Now we are going to talk about their atmospheric requirements. So the atmospheric requirements also groups bacteria into various forms. So we have the obligate aerobes. We have the anaerobes. We have the micro aerophiles. Okay. We have the micro aerophiles.